Recording. Now, where is this one right here? That's what I want. So, I hope you guys are doing all right here on this lovely Saturday morning. Open up my window here. Nice day, get outside. I've got my two kids downstairs. My missus is working, so we're just busy morning here. And I'm recording this for this group here, Q, and I'm also going to be recording this there for the other group there that I have that is R. And I've updated the PowerPoint, by the way. I've been working on this PowerPoint there, and I just didn't like the previous PowerPoint, so throw that one away. And I have updated the PowerPoint there. It's much better. All right, and so what do we got here? We've done week 1, 2. This is week 3. Okay, today we're going to be dealing with, we're going to start some microbiology. We're going to introduce you guys there to the bacteria, and it's kind of like a two-parter. Like there's this week here is bacteria. Next week there we'll finish bacteria. We'll get into population and disinfection and all these kinds of things, and we'll do viruses.
But I'm here.
Oh, hello. Hi, guys. If you have a question, put your mic on. If you guys could just mute your mics, that would be wonderful. Hi, Jonathan. Hi. If you could turn your microphones off, that would be wonderful. Okay, Jonathan. Okay. Not seeing it here.
Jonathan, you better go upstairs and use mine. You're so freaking stupid.
Hey guys, if you're not listening, you need to turn your microphones off. There's
Jonathan, you better go upstairs. This is so freaking stupid.
This is kind of hilarious here. Please turn off all microphones. Please. There we go. Okay, I hope everything is all right now. Thank you. This week here again, we're doing bacteria, and it's good stuff. You don't have to have a huge microbiology background, okay? And it's okay. Yeah, it's all good, Monica. We're just having fun. It's Saturday morning, and everything is good. So this week there, we're going to do bacteria. Next week there, we'll finish bacteria. Like you can really do bacteria in a week and a half, 2 weeks. You could spend billions of years doing bacteria, which is what exactly they've done. And they're quite amazing. Hence the reason that we have a course like this, because they've been around for billions of years and they're efficient at what they do. And yeah, it's going to be interesting. Next week there, we have viruses, bacteria, the prions, which are weird ones. I still hear microphones on. Can everybody check that their microphones are off? Thank you. Okay. I may have to say it again. We'll see. The prions, viruses, I've got a whole bunch of videos there for next week. And apologies on the lateness of getting the PowerPoint out there. I'm making this course. I have the syllabus and all that, but I like to make things my own. And in order to do this week, I have to do next week, I have to do the quiz, and I have to do the test all in one shot. Because I sent the announcement out there. This week there, we're doing bacteria. Next week there, we're going to do bacteria, viruses, prions. We'll do a bit of fungi. Just introduce you guys to the microbial world. A little bit of microbial control. And after your class next week, okay? So I'm thinking there that 12 o'clock for everybody, the quiz will open. And I think you'll have a week to do it. I don't know if I'm going to leave it open for an hour. Like the quiz will be there, and you'll have a week to do it. And I think you'll have 2 or 3 minutes of question there, so you guys will be fine. Then we have thanks. We have what, week 3, week 4, and then we have Thanksgiving. So there is no class, and then week 6 I think is test 1. Okay, and there's no class there that week. It'll just be a test. Basically, it'll be double or triple of a quiz. So I'm thinking there's somewhere between 50 to 60 multiple choice questions. And so nothing too crazy. There's no tricks. There's no anything for this course here. That's where we are. And yeah, do you guys have any questions there for me? If you have a question, you can turn on your microphones. But I think that we're on track. As I said there, week 1, and this course here won't be. It's not going to get you. All right. And if it seems to be getting you and irritating you and all these kinds of things, get a hold of me and we'll get you guys all straight. Okay. It's basically a PowerPoint based test, and yeah, that's where we're at. How we doing? There's still microphones on. Okay, and if can everybody for the whole class, can you guys make sure that your microphones are? I'm just gonna go through. Josian, can you please turn off your microphone? Do we have any others here? Jonathan Ho, can you please turn off your microphone? Okay, very good. Hello.
Hi, this is Anna. You can actually mute them.
Can I?
Yeah.
Oh.
I just don't know how to do it in mine, but you can.
All right, thank you, Anna. I'm going to do that. Very good. I'm going to, yeah. I'm, as I said, I'm more of a WebEx person, but I'm going to see if I can, microphone real time. No. Same as me. Test. I'm going to figure out how to do that. But anyway, we'll get going. It's not too bad. If someone has their microphone on, I can totally live with that. But thank you, Anna. So, without further ado, do you guys have any questions there for me that are emergencies or anything there that you think that the class should hear? If not, see me after class or shoot me an email. And yeah, I think we're doing all right. Are we good? Rock and roll. There we go. Now, I originally labeled this there bacteria and viruses. Oh, this is the wrong one. Sorry. Okay, very good. That's what we got. I'm just making sure this is the right one here. Yes. No, it's not. What is happening here? That's what I'm looking for. Where is it? Actually, yeah. Week 3. Bacteria. This is what I'm looking for. Whew! Very good. So, here we are. Bacteria basics. And as I said there, that you guys can get a PhD in this. And that's all right. I just want to make sure there that my... what's going on here? Why won't that just let me open up? Okay, so you guys let me know if there's any issues. I can't see my the comment section, so if there's anything there you guys didn't get, just let me know. And I changed this PowerPoint up there. I was just looking at this there, and again, this is the problem with the VR course, is that I don't know as much about you as I would like. And so I just thought I'd take it back a step, and let's just go into the characteristics of life. And I've got 10 here, and like we're talking about the definition of life. We're talking about, is this yellow highlighter, is it alive? How do we prove that it's not alive, right? Like, this is scientific thinking, scientific thought. Okay? And, okay, let's see here. Where's the chat? It's me, John. I'm not able to hear you. Hi, Jonathan. I don't know what to do. You'd need to unmute your computer. And I will. Hi, Ellen. I will. The new PowerPoint is posted, but I'll make sure that it's posted there. And if it's not, then we'll get it up there. Like the whole idea. What is life? And to be alive, there has to be movement or change in position. Like, trees don't really move, but they have the ability to move. Unless you're an Ent from Lord of the Rings, where you can get up and walk around. All right. Responsiveness. You've got to be able to react to your environment. If there's a change in weather or something or this or that, can you adapt? Can you change? Okay. Growth, increase in body size. Okay. Circulation, movement of fluids around. Okay. Doesn't matter if you're alive or, if you're alive, you've got to have water, right? Moving things around. Reproduction. Respiration of some kind. Maybe not oxygen. There are certain bacteria which we'll see that use sulfur. And if you'll see where sulfur's position on the periodic table is, it's directly below oxygen. So in a lot of ways, sulfur can act like oxygen, and some bacteria that live at the bottom of the ocean near volcanic vents, that's what they use. Interesting stuff. We'll follow up with these. Assimilation. Changing of absorbed substances into chemical. Digestion, breaking down of food. Excretion, absorption, things passing through membranes in both directions. Okay? And is it limited to this? No. But these are just some of the things there that living things do. And you have to think, like next week there we'll do viruses. And do viruses viruses move? Yes, they do. But, do they grow? Yes, they do. Do they reproduce? Yes, they do, but not by themselves. They go into a foreign cell and commandeer its ribosomes, and ribosomes, which we'll get into there today, are protein makers. And so a virus will come in and invade a foreign cell, commandeer that cell's ribosomes, and start making protein for itself. And in that way, that's how it reproduces. So, you know what I mean? There's lots of things here. Things that make you go, The maintenance of life. Water, food, oxygen, or some type of atom there that's able to hold on to electrons for the purpose of extracting food or energy from our food. Heat, okay, and pressure. Funny enough, if you got to have pressure. How do trees know how to grow up? It's because there's pressure coming down, right? And so they follow the opposition of the pressure, right? Life characteristics which obtain, release, and utilize energy are largely part of a metabolism. And metabolism, this is just words there that we use to express all type of chemical reactions going on in the body. Okay? Metabolism. What do we got here? Requirements there. And this just follows up. Water, food, oxygen, heat, and pressure. And y'all can have a look at these there. But these are things there that organisms need. Now, organisms. Is a virus an organism? Wait until you see a prion. It's really not even anything. Just a bunch of protein strands come together. Interesting stuff. But we'll get there next week. And just the whole idea of microbiology, and that there's what's the word I'm looking for? There's a whole nother universe out there, and it's right on the It's right on the tabletop right in front of you right now. There's a whole world going on. And just to not skip ahead, but I just wanted to show you guys here. Like this is electron micrograph of a pinhead, right? So this is really small. Okay, this is a pin, right? And you'll see here there's continuing zooms up. You know, we're zooming in here, and you can kind of see that there's stuff on this pinhead. You zoom in even further, okay? And I am going to zoom in. And you'll see them all right here. Oh, sorry. You'll see them here. And you'll see there that this pin is absolutely loaded with bacteria. As most things are, they're all there. And you can't see them. And, you know, now that we have, you know, scanning electron micrographs that can see these things, we have all the technology there that we could ever want. You think there 500 years ago, you know, the microscope was just being developed. You know, actually not even 500 years ago, probably about 350. And, you know, how do you know that there's bugs there? They suspected that things were coming, or things were there that they couldn't see. And then Louis Pasteur, I think in the mid 1800s, 1862, don't quote me on that, but came up with this, and it's the whole idea of this spontaneity, spontaneous creation. Because they knew that if they left, and they use this term a broth, so let's call it chicken broth, or I don't know, some type of meat broth or whatever. And it's a nutrient broth that they called it. And if they left this exposed to the air, and you see here, you have a look here, the first experiment, here I've got boiling, so we sterilize this nutrient broth, and the flask was outside the building briefly. Okay, and so they took this, the top off here, and so they cooked this, sterilized it, left this outside for a little bit with the top off, and 89% of the broth, of the broths bottles there that they used, demonstrated bacterial growth. And then what they did was he again boiled nutrient broth, and the flask was inside the building and opened briefly. So only 32% of these ones here developed bacteria. So it's like, if I take this outside, there's more of a chance that the bugs are going to get in here. But if I keep this inside, there are still bugs inside, but not as many. Okay, conclusion, hypothesis B is supported because relative concentrations of bacteria in the air explain the results. More bacteria in the air outside than inside. All right, let's look at the second experiment here. Here we've got a boiling flask that's got a wonky tube on it, right? There's a bend here and a bend here. Well, they boiled this broth there to neutralize it, and they left the flask open to the air. Okay, and then afterwards they realized, they tested the results, that the 100% no bugs in here, and 100% no bugs here, but where the bacteria seemed to collect was right here. So we knew there that the bugs are getting in here, but are not getting up here. So, you know what I mean? There's little things like this that, you know, if you don't know what the bugs are, do an experiment. If you think that something's happening, see, they're suspecting that something is getting into this broth, and they don't know how. And through scientific thought, through scientific action and practice, you know, you can come up with this, and they realize there that there is something out there. Now, maybe at this point in time, Louis Pasteur didn't know, we didn't have a name for it, these kinds of things, but there's something there. There are things that we still do not know. And even to this day, more so, in fact. Okay? The more we realize, the more we learn, the more we realize there that we don't know as much as we thought we did. Welcome to it. Bacteria are microscopic, unicellular, one-celled organisms that lack a nuclear membrane. And we're gonna get into all this. I'm more introducing this. I'm actually gonna take you back and oh, I see 9 chats. Is everything okay here? Try clicking the MDR. Okay, so Jonathan. Okay, so Jonathan, you're sorted. Okay, all right. So if there's any issues, okay. So if there's any issues, let me know. I hope you guys can gain access. If there's any issues there, I'm also gonna post this there to YouTube as well. Okay, so anyone who just joined late, we're just starting bacteria. Okay, your quiz 1 will open next week. Okay, after class, I'm thinking 12 noon. It'll be open there for a while. I'm thinking there for the entire week. And then the week after, okay, week 4, and then your quiz will open immediately after week 4 at 12 o'clock. And then we have Thanksgiving there the next week, so there is no class week 5. When you guys come back there, week 6, there will be a test. Week 6, no class, just test. Okay, and I'll probably have week 6 test operating there during class time. That's what I'm thinking. It is a test, right? Okay, so bacteria can live on land and water in the air. Bacteria live everywhere. Okay, and they also live in and on both living and dead plants and animals. Like they're just literally everywhere. And I think there last week there I was talking about there's a big thing there with global warming, that you have these bacteria and bugs that have been trapped in the permafrost up in Canada and Russia's north for the past 100,000 years, and now they might be coming back. So it's interesting to see what'll be out there. Most bacteria are heterotrophs. Heterotrophs are organisms that cannot make their own food. Perfect. All right. So we just showed you guys there a pinhead, and there's lots of bugs on here. So if you pricked yourself with this pinhead, you would be infected. That's why these are so good evolutionary. Okay, these guys have been around for billions of years, and they will continue to be. No way we'll ever be able to get rid of them all. And you'll see that I've got slides after slides after slides. I'm a big fan of if you don't like if one slide doesn't catch your fancy, well then I have another one there that because everyone learns in different ways. Okay, so the bacteria come in all sorts and shapes and sizes. Okay, we've got these spheres, and we call these guys cocci. And you would have heard of them, staphylococci. Okay, and these names, they come from partially their size. So you've got cocci, which are these guys here, spheres. You've got bacilli, which are rods. Okay, you've got spiral. Look at these guys here. They're wiggly little worms, all right? And so you've got these ones here. I've got lots more pictures here. And you see the, look at these cocci here. They kind of look like grapes, right? They're in bunches, one on top of each other. And then you'll have ones there that are in straight line. These are still cocci, but they're in straight lines. Okay? There we go. There's some bacilli. You see these guys here? Jeez, man, it kind of looks like my all-brand cereal. I won't be eating that anymore. All right. And here are the some spiral bacteria here. Just some shots, okay? And you remember there's your cocci, your coccus. These are the round spheres. Diplococci. Dip 2. Okay. Staphylococci. They're the ones that look like kind of like grapes in bunches. Streptococci. These cocci here are in lines. Okay. You've got them. You've got tetra. And you know what I mean. There's lots of them out there. And you've got, here's your bacillus. Kind of rods. Diplobacillus. Okay. These guys, this is called palisade. Okay. Streptobacilli. Okay. Streptococcus. Streptobacilli. Strepto means in line. Okay. And you just have a look at these. And, you know, if you didn't like that slide, well, have a look at this slide. Okay. A is your bacilli. B is your coccus. Okay. C and D. E, what we call spirillum. These are the spirals. F, these are your vibro. Okay, more like comma shaped. Okay. Anything crazy there, let me know. And just to show you again, kind of the same slide, but I like this one here because it shows you in comparison there to an RBC. Okay, there's your RBC in the top. Okay, and there's your size comparison. All right, that's where these guys are at. And you'll notice there that we're dealing in microns. Okay, there's 1,000 microns in a centimeter. Okay, sorry, a millimeter. All right, these are 10 to the negative 6 meters. So, cell shape is determined by the genetic character of the organism. Its genes code for the synthesis of the cell wall material, and the cell division mechanisms there that result in a round or rod or spiral shape. We'll get into this. I'm not really interested in you being able to possess bacterial taxonomy, being able to name bacteria. No, that's not the goal of this course. If that was the goal of this course, I would need to have you in the lab. Okay, and but being familiar with bacterial identifiers, absolutely, and that's what we're going to be dealing with here. Okay, and next week there we'll deal with more into relating a bacteria to a disease kind of a thing. So today all you need to do is just sit back and relax, and we're just going to go through these things, and at the end of it there, hopefully there you'll have a more general understanding of bacteria and where they are in your universe. Okay, but before we do that there, we got to go back, and we're going to talk to you there about eukaryotes, and eukaryotes as opposed to prokaryotes. And you'll see there that bacteria are prokaryotes. Okay, they're simple. They're very old. They're much older than us. Okay, and they're very simplified unicell organisms. Okay, and we'll show you these, and we'll show you all the things that are going on. A eukaryote, that's us. That's a salamander. That's a blue whale. Okay, higher, more high. I don't want to say higher level, but more specialized. Yeah, have a look here. Eukaryotic cells here, we actually have a nucleus. And if you don't know what a nucleus is, I'm going to go through, and we're going to go through all these things right here, okay? And at least the important ones, because this is part of the course here. I'm not going to go after all of these. It's impossible. How can I do that on a 30-question quiz or a 60-question or a 50-question test? I can't. You have to think the amount of information there that I've given you so far is crazy. I could write 10 tests from it, but I only get to write one. Okay, so when you guys are studying for your tests and these kinds of things, sure, know everything, but that's an impossibility unless you have a photographic memory. And again, photographic memory doesn't mean that you've learned it. You're just able to recall it. Okay, but knowing everything, no, pick and choose what the most important things are, and you'll see where we're all at the end of this. Okay, the DNA is membrane bound. Okay, and you just have a look here, and this is a eukaryotic cell, and there's lots going on here. I'm just going to, can I move this over here? You're just going to let me do it. There we go. And you'll see here, we'll start off there with the nucleus. Okay, there's all this stuff, and a cell, like this could be a cell in your big toe. Okay, and you'll see there that there's a lot going on. And if you want to think of the nucleus as the brain of the cell, I don't mind, it's not. Okay, it just stores all of your genetic information. So basically it's like having Google, that if the cell needs something here, hey nucleus, Give it to me. Okay, so the nucleus will read part of its DNA, print off, okay, a flimsy fake copy of a certain section of the DNA. It's called mRNA, which travels out, and it'll go to the what we call the ribosomes, okay, where a protein is made, and it's the protein there that the cell was looking for. But we'll go through all this here. You've got the nucleus, okay, your nuclear envelope here, and you'll see these pores here. This just allows things like mRNA and all these things to come in or to come out and all these kinds of things, okay? So this is where the DNA is stored, okay? You'll see here there that we have endoplasmic reticulum, okay? You'll have, yeah, we're showing you guys rough endoplasmic reticulum here. And basically, you'll see little black dots on here. These are all the ribosomes. I'm just trying to find, where are they? Yes, right here. Sorry, I missed it. Ribosomes. These are the little ribosomes here. And when we're going to be talking about the ribosomes more and more there this week and next week, because these are the little guys here that make proteins. And I'll show you how they work there. There's a slide or two there. And like when some proteins are made here or whatever the process is going on, what'll happen is the DNA will get copied, okay? And this copy is mRNA, which will float outside, and it'll go right to one of these ribosomes here, where the ribosome will translate this mRNA copy of the DNA into a protein, and it's this protein there that the cell needs at this particular time. Well, when you're talking bacterial or you're talking viral, like especially viral here, if the cell gets injected there with viral DNA or viral RNA, what happens is now these ribosomes start this, instead of a cell working for you, now this cell is working for the virus. Okay, and yeah, it's interesting stuff. It really is. Okay, and we're just going to go through all these here. The membrane-bound organelles, the package where specialized functions take place, like mitochondria. These are the powerhouse of the cell. Okay. So this is where the energy comes from. You'll see these little guys in here. They're called cristae. Okay. This is where the energy for the cell comes from. Okay. You'll see here that there's no eukaryotes there. We don't have cell walls. We're not like plants or bacteria. Okay. There's a cell membrane. Okay. Which is porous to some extent. Some things can come in. Some things can't. Okay. There's your nucleus there, your nuclear envelope. Your DNA, your chromatin is in here. Okay. There's your rough endoplasmic reticulum. Ribosomes here. Okay, smooth endoplasmic reticulum here. Golgi apparatus here, they're talking about for packaging. Okay, because the membrane allows certain things in or out. Okay, some things can just move freely in and out. That's all good. But some things like protein cannot. So any type of protein there that's made, coming in, coming out of the cell has got to be packaged up. All right. There are microtubules, and you really can't see these, but this is basically the scaffolding of the cell, and it keeps the cell that shape. Okay, so it's almost like an internal skeleton, but it's not a skeleton. Okay. Yeah, centrioles, peroxisomes, lysosomes there, these guys there, peroxidase just for degrading, cleaning up, there's a garbage system in here. And there's little bits of floating bits all over the place there, and we got to keep things moving. We got to keep things cleaned up. Okay. So there's your, there is like a eukaryotic cell in a nutshell. You'll see there that there's, I'm just going to leave that and we'll just keep on going. All right. So the cell wall, if present, okay, is chemically similar. Simple there. We're talking cellulose, okay, like for plants there. Slightly larger ribosomes. Cells divide by a process like mitosis. Okay, skin cells, somatic cells, they're the body. Somatic cells, body cells, they'll divide there through mitosis. Okay, so you get exact copies. But then you have meiosis. These are for the sex cells, the gametes. We talked about this there. That's why a person can have children with the same spouse, and the 4 children will look completely different from each other. All right, it's all has to do with genetic variability, and we don't want any 2 individuals in a population having the exact same DNA, because then they are now susceptible to exactly the same diseases that may arise. Welcome to all these guys. Okay, well, welcome to COVID. All right, any questions? Let me know. Is everything going okay? I hope so. I hope you guys are doing all right. So, eukaryotes, nucleus with a nuclear envelope, bigger, more complex than prokaryotes. These are your bacteria, okay? Membrane-bound organelles, okay? DNA is double-stranded, 4 chromosomes, fairly highly organized. Can be uni or multicellular. Okay, there are bugs out there that are unicellular, multicellular. Animals, plants, fungi. Okay, those are what we call eukaryotes. Prokaryotes. No nucleus, no membrane-bound organelles. The only one there, just the ribosomes. And there's that ribosome again. Those are the protein makers. Okay, which bacteria have, but they're smaller, not as complex as ours. All prokaryotes are unicellular, one-celled, and they're very old. They're very, very old. They actually, there's a theory that you'll see there a nucleolus there. You'll see that they think there that some point in time, billions of years ago, or a billion, million, I don't know how long ago, that a eukaryotic cell actually digested a bacteria and incorporated it and made it into its own. So there's lots, because in our mitochondria here, it has its own, I want to say, bacterial DNA, plasmid DNA, and it's our DNA, but it's interesting. It's round DNA, circular DNA, but that's all right. We'll get in there. And again, where I'm at with this course is, I don't know where you guys are in your biology. I don't know, and what I'm doing is I'm kind of like throwing a stone on still water. We're just kind of skipping the surface, okay? And that's what I'm looking for. I'm not looking for a PhD in microbiology at all, okay? I'm not looking for anything. We're just going through this, and the slides are what we're looking at. This is the forerunner there to the eukaryotic cell, as the theory goes, because the prokaryotic cell, the bacterial cell, is very simplified as opposed to the eukaryotic cell. As you see, there's your eukaryotic cell. Okay, there's a lot going on here. Okay, you've got a vacuum system, you've got a brain system, you've got a power system, you got a packaging system, you got a protein system, you got a transport system in and out. There's a lot going on here, and this is all moving and shaking all at the same time. All right, and you have a look here. Here's your bacterial cell, typical. Okay, this is obviously bacillus, but you can see there's really not much going on, and we'll come back to that in a second. A single closed circular chromosome contained with the nucleoid there. Okay, there are additional circular DNA called plasmid DNA. Okay, you got ribosomes, again, for protein synthesis. There's an outer cell wall reinforced with peptidoglycan. I like that word, peptidoglycan. This is the chemical compound that's found in the cell wall that gives the bacterial cell wall its toughness. All right? And in a lot of cases is where the antibiotics attack. They'll attack the cell wall, okay? And that's where they'll go. That's how an antibiotic will actually destroy a bacteria, okay? Some of them anyway, okay? And for some bacteria, they have a flagella, okay? We'll get into all these kinds of stuff. And again, you'll see that I've included a couple of different slides. I've gone through my micro library, and it might be overkill, but maybe you like one slide over another, and that's a slide that'll get you through to a 95. What do I care as long as you get your 95, right? Okay. So bacteria are prokaryotes. There's no nucleus or organelles per se. Okay. DNA is a singular circular chromosome. There's no histones. Now, if you don't know what a histone is, it's a histone are these little, I don't know, call them balls, and they're just hard balls that are found within your DNA and within the chromosomes, and it's what the DNA uses to wrap itself around and bend and turn. It's the histones there that give DNA its kind of solid nature. Okay, and you'll see there that for bacteria, they don't have these histones. And you look at their DNA, it's all just mashed potatoes. Okay. The cell walls of they contain peptidoglycan, which is a polymer. Okay. Well, I have some other stuff there. We'll read there. Okay, about that. It has ribosomes, but they're smaller, so there is protein synthesis going on. And you'll see there that the bacteria do not divide, okay, like we do in mitosis. They divide through something there called binary fission, which I'll show you. That is good coffee. Another bacterial diagram, and you see here, if you didn't like the last one here, okay, there's the mashed potatoes. Okay, there's the nucleoid there. There are your ribosomes. There's your membrane on the inside. Okay, your cell wall. Okay, there's your membrane there. Your cell wall. And your capsule there. These are called pili. And this is what a bacteria uses there to stick somewhere. Okay, it's got these kind of like a Virginia creeper vine, you know, it's got these little tentacles there, and that's how it uses to adhere to a surface. Okay, and again, there's not much going on here, right? And in some cases there, not all of them, but in some cases there, some of them there will have a flagella for moving around. Okay. And again, kind of the same slide, but each slide here has different things there that I want you guys to notice there. Okay. Glycolax there, you've got a coating or a layer of molecules, okay, to the external cell wall. Okay, and it's on the, there we go. Bacterial chromosomes, nucleus, there you go. There's your pili, okay, or pilus, an elongated hollow appendage there used to transfer DNA. Okay, there's your pilus. Okay, fimbriae, these are these hairs there that help you there to stick to surfaces like that pinhead. You've got your flagellum. You've got all these guys here. Okay, and just to show you guys there that there's really not much going on. Okay, and I like this slide here. This is my favorite of them all. It's simple. It's clean. That's how we do it. Okay. And here's another shot here. And I use this one here because I like this. It demonstrated there the mechanism there of this flagellum. And you'll see how it all moves and how it attaches here and this and that. There is some hierarchy here, but you'll see there the simplistic level of these bacteria. And they don't need to have higher level function. They're doing just fine on their own. Okay? And the only reason we've been able to keep them at bay for the past 100 years is for antibiotics. And next week there, I'll go into a little heavier. Every week I go into a rant about antibiotics and doctors and these kinds of things, right? Well, I'm not going to do it this week, I promise. I'll do it next week in a little bit more detail. Okay? So that's what we got going on there. And another shot here. This one here is another one as well. And again, it has all these things right here. And again, we're going simple. Okay. So DNA, ribosomes, flagellum, pili, cytoplasm. That's where we're at right now. Okay. The bacterial appendages there, the pili, or also called the fimbriae, okay. There's a flagella, okay, that are short hair-like proteins, sticking to surfaces, okay, very hard to wash away, okay, these kinds of things there. And I did this there. I saw this there in one of my slides here. If you break a prokaryotic cell down, so a bacterial cell, okay, you've got the cell envelope. Okay, there's your glycolax there. Okay, your cell wall and your plasma membrane. Okay, your peptidoglycan is in here. Okay, and this provides there the protection and maintains the shape. Okay, you've got the cytoplasm. And the cytoplasm is the fluid matrix there that's in our cells and bacterial cells. Okay, it's just basically the water solution that the cell is contained with. Okay, and this allows things to move, proteins to flow in and out, mRNA to move, and these kinds of things. Okay, there's your nucleoid, your ribosomes. Don't worry about thylakoids. Okay, you'll see a little bit of cyanobacteria there next week. I go into about this much detail on some bacteria there and what they do. It's next week's slides are awesome. I actually wanted to show them, but we'll be here for 4 hours. The appendages there, the flagella, the fimbriae, the pilus there, and we'll show you guys more. Okay, so just an interesting way to break a prokaryotic cell up. There really isn't a lot there. Okay. Just another shot there of the flagella. Okay, and you'll see there, I'm not going to go into the S ring and the M ring and the mechanics of this. I just wanted to show you there that there is a locomotion center. Neat stuff. Okay, I'm not interested in monotrios and these guys here. I just wanted to show you there that there is this mechanism involved in the flagella moving. Interesting stuff. There are some similarities, though, between eukaryotes and prokaryotes. They contain all four biomolecules. You've got lipids, which are fats, carbohydrates, which are sugars, okay, protein, which is protein, and nucleic acids, okay, DNA. Some DNA, and when we wait until we get into viruses there, things get a little wonky. And you remember your whole definition of life? Apply it to things. Apply it, when you're just out walking around, I gave you guys 10 definitions of life. Start applying them there to these things. Deductive reasoning. Here is an old baseball cap. Okay, can you apply your definition of life to this baseball cap? Can you do it to this? Deductive reasoning. Start questioning the world around you. Okay. They both have ribosomes, both have DNA. Similar metabolisms, just the whole idea of consuming and excretion. The whole idea, getting energy is getting energy. Okay. Eukaryotes can be unicellular. Okay, you've got microbiology, mushrooms. Yeast is a unicellular fungi. Yeast, good. They have cell plasma membranes or a cell wall. And again, I could spend weeks on this very slide, months, but I won't. And this, sorry for it looks like mashed potatoes, but I just wanted to show you, comparatively speaking. And are we going to go crazy? No, we're not going to go, but this was a kind of a nice slide here on what a eukaryotic cell looks like. There's your nucleus, okay, there's your ribosomes here, there's your rough ER, as we say, endoplasmic reticulum, your smooth endoplasmic reticulum, and, there's your ribosomes here. There's lots going on. You see the hierarchy. You see how complex one of these cells are. Okay? And then, here's a plant. And there's a lot of similarities here. Okay? But plants there, they have chloroplasts. Okay? Chloroform. chloroplasts, all right, and that's what they use there to make energy there from the sun. But you'll see there they have a nucleus as well. The big difference there with plants is they have this thick cell wall, cellulose. That's why you can somebody takes their car and smashes it into a tree. Well, the tree's not moving because cellulose is very, very hard substance, right? But you see there's a plant, okay? And then you see a little wonky little bacteria. There's really not much going on there, okay? There's your ribosomes in here. This is all your cytoplasm. There's your nuclear material, cell wall, plasma membrane, pili, flagellum. Really not much else. So, but as I said, these guys don't have to be. These guys were probably one of the first organisms to be made. Sorry, there's a wasp that just got in. Sorry, buddy. Okay. You got whacked. All right, good enough. But they don't have to be because they're such efficient at what they do. Okay. Here's another shot here, just very simplified. You've got a eukaryote versus a prokaryote. Okay, and you'll see there the differences there. The prokaryote, the bacteria, the DNA is kind of free floating. It's not in any type of nuclear membrane. There's a capsule there and a cell wall. There's really not much there. Okay, neat little slides. That's what we're doing there. Any questions? No? Everything's good? What do we got? Two types of DNA there that can be found. Okay, you got bacterial DNA, but then you can also have the plasmid DNA. Okay, and the plasmid there, what we call extrachromosomal DNA, is not part of the genomic. Okay, it's more disease and defense. Okay, for genes there. Okay. And it's just a tool that's been developed over billions of years, and it works. It works. These are the shots there of the ribosome, and the ribosome has two subunits. And what'll happen is the in a cell and you guys can go on YouTube and see just Google ribosome function. Google ribosome function there on YouTube, and this will come on up. There's a million videos, and the DNA, a certain section of it will get copied, and this copy is called mRNA. It's called RNA, mRNA, messenger RNA. And what happens is this little copy of the DNA travels outside of the nuclear membrane. Okay, well in bacteria there isn't any, and it travels over to a ribosome. And what happens is this string, this copy of the DNA, it travels through kind of like a stock ticker tape. And it travels through, and every section of the mRNA that gets read, okay, by the ribosome, what happens here, an amino acid gets put onto here, 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 here. And as this ticker tape travels through, this protein gets longer and longer and longer and longer and longer. And so that's how your body makes protein. And protein there is involved in all sorts of things. Okay? Uh-oh. Are the kids coming up? Here. That wasp, man. Uh-oh. The kids might be coming up. Shh. Maybe they won't know we're here. So inside the bacterial cytoplasm there, you got the ribosomes, okay, cytoprotein synthesis. The only difference is they're smaller than a eukaryotic. Okay, that's all we're like. And just to let you know that remember we were talking about antibiotics and how some antibiotics there they attack the cell wall. Well, there's other antibiotics there this is where they attack. So the antibiotics there, they don't stop the bacteria from invading the cell or whatever, but what will happen is the antibiotics there will stop with this process. so that the bacteria can't make protein. And if the bacteria can't make protein, then therefore now it is now basically defective, defunct. It will die. Okay, and so two places there that antibiotics can attack: the cell wall or the ribosomes. Okay, this is just an electron micrograph there of bacterial chromosome. Pretty exciting, eh? Good. This is bacterial dividing by what we call binary fission. Okay, not the same as what we do. Okay, or eukaryotes, but let's just go through it here. So you got the prokaryotic chromosome. Okay, plasma membrane, cell wall. Basically, you've got some ribosomes in here and some pili and a flagellum. That's it for a bacteria. Duplication of the chromosome. So we're doubling. Okay. Continued growth of the cell, and you'll see there it starts to divide. Boom. That's it. That's what we call binary fission. Okay. And that's what it does there. That's how it divides. Nothing too crazy there. Bacterial vocab. I've got a couple slides here, and here's some good words here that I'd like you guys to know. Just, a question on a test. Okay, some bacteria can undergo conjugation using sex pili. Okay, conjugation allows for the form of sexual recombination as genetic information is exchanged between bacteria. So there is some type of sexual production here amongst bacteria because they also need to exchange genetic material as well. So they can just double themselves. Okay, easy peasy. But there is, during conjugation there, they can exchange genetic material, which is their way of producing variant strains of bacteria. So, all right, and that's how they protect themselves. Genetic variability, same as us. Bacteria can undergo transformation when they incorporate DNA from the environment. Okay, so when they encounter DNA from the environment, they can just incorporate it into their genome. That's pretty scary. Transduction. When bacteriophages transmit viral DNA from one bacterial cell to another. Okay, so the process there of moving bacterial DNA from one to another is called transduction. When bacteria experience unfavorable environmental conditions, they can form spores. These are, let's call them an egg, let's call them whatever. So you have your bacteria, and if it becomes too dry, and okay, man, this wasp has got to get out of here, dude. Sorry, dude. Wrong time, wrong place. There we go. Got him. Now, these spores, when it becomes too dry or the conditions aren't good and the bacteria, it can't reproduce, what happens is it goes into a spore form. Okay? And these spores can remain dormant for thousands of years. And the whole point is not for reproduction, it's for survival. And what happens is the bacteria will make these spores. And these spores there, this is one of the big problems, is that we can kill bacteria with 70% alcohol. We can do all these things. It's the spores that are the problem. These are the ones here that are highly resistant to environmental stress and can be dormant for months, years, thousands of years. Okay, and what these guys here is they're not eggs. And when the time is right, okay, for them to germinate, they will. And now they're good to go. And from this, we'll make a bacteria. And then from one bacteria, we'll make 2, 4, 8, 16, 32, 64, 128, 256. All right. Guys, what we're going to have to do is we're going to have to stop it here. And I got to go and take care of my kids. It's an hour anyway, so let's go. Let's take 10 minutes. I'll see you guys back here at 10:10. How does that sound? Is that okay, guys? All right. So, all right. Well, I'll stop the video and I'll just pause this. So I'll put it on mute. I'll stop the video so the link will not be broken. But we'll see you guys back here in 10. Okay. Thanks, guys.